Hello, good afternoon. Welcome and thank you for joining us at this inaugural Distinguished Professorial Lecture by Professor Duraisamy Saravanakumar, Professor of Plant Pathology at the University of the West Indies, St. Augustine Campus, Trinidad and Tobago. The topic of his lecture is Microbes, a Devil or a Savior of Plant Health. I am Professor Neela Badri and I am the Chair of the Open Lectures Committee at the University of the West Indies, St. Augustine Campus. I would like to acknowledge the presence online of Professor Brian Copeland, Pro Vice Chancellor and Campus Principal of UWI St. Augustine. Professor Duraisamy Saravanakumar, our distinguished Professor of Plant Pathology from the Department of Food Production, Faculty of Food and Agriculture. Dr. Mark Woodivera, Dean of the Faculty of Food and Agriculture, University of the West Indies, St. Augustine, and Dr. Gaius Eudoxy, Senior Lecturer and Deputy Dean of Outreach and Internationalization at the Faculty of Food and Agriculture. Heads of departments and members of management of UWI St. Augustine, staff and students of UWI and from other universities, family and friends of Professor Duraisamy Saravanakumar, in particular his spouse, Dr. Ramya Saravanakumar, and his two lovely young daughters, Ruth Vika and Litika, our specially invited guest, the public and members of the media. Again, thank you for joining us at this distinguished Professorial Inaugural Lecture. One of the aims of the Open Lectures Committee at the St. Augustine campus is to create opportunities for newly promoted professors to present their inaugural professorial lecture and to present an overview of their contribution to their field, their academic peers, students, collaborators, and the wider community. The topic of today's professorial lecture by Professor Saravanakumar is quite exciting, since plants and plant health are important to our survival and well-being. Microorganisms play a critical role in the management of plant health. However, they can also deteriorate plant health and cause diseases affecting crop yield and agricultural production. Plant diseases ultimately attract the application of synthetic pesticides, leading to human and environmental health concerns. Professor Sarah, or Professor Saravanakumar, and popularly known as Professor Sarah, has conducted groundbreaking research in the development of biopesticides for the management of diseases, attributing to sustainable crop protection. In other words, microorganisms can serve as biocontrol against plant diseases in controlling the plant pathogens, and the bacterial biopesticides tend to be target-specific, rapidly multiplying, easy to handle, non-toxic, economically suitable organisms, and better survival and longevity. This afternoon, we celebrate with Professor Sarah his achievements through the advancement of his knowledge and excellence in teaching, research, innovation, intellectual leadership, and outreach. He has achieved the highest academic rank of professor at the University of the West Indies. Congrats, Professor Sarah. On a personal note, I first met Professor Sarah when he joined the Department of Food Production in 2014. At the time, I was serving as the head of the Department of Food Production. We were very fortunate to have attracted such a distinguished academic to join the University of the West Indies. He had an impressive resume. I was the only microbiologist at the time at the Faculty of Food and Agriculture, but with Professor Sarah's expertise in plant pathology, microbiology, genetics, and molecular microbiology, has contributed significantly to the advancement of the discipline at the Department of Food Production, Faculty of Food and Agriculture, and to the broader University of the West Indies. He's now serving as the head of the Department of Food Production. I will now like to invite our campus principal and Pro Vice Chancellor, Professor Brian Copeland, to bring some opening remarks on behalf of the University of the West Indies, St. Augustine. Welcome, Professor Copeland.
Thank you, Professor Neela Badri, Chair of the Open Lectures Committee. I'll go right into my presentation and say a good afternoon to Dr. Mark Woodivera, Dean of the Faculty of Food and Agriculture, Professor Duraisamy Saravanakumar, Head of the Department of Food Production at the faculty, Dr. Gaius Eudoxie, Senior Lecturer and Deputy Dean of Outreach and Internationalization of the Faculty of Food and Agriculture, family and friends of Professor Saravanakumar, ladies and gentlemen, members of the media, good evening. On behalf of the St. Augustine campus of the University of the West Indies, I am pleased to welcome you to this Professorial Inaugural Lecture by Professor Duraisamy Saravanakumar. He will this evening speak on an area that may be obscure to many, a microscopic world that affects our very lives. I think we all agree that the pandemic has been disruptive. In its early phases, it forced people across the world to remain indoors for many months in an effort to combat an unseen enemy. Global supply chains began to weaken. But we've also seen how that very forced isolation also caused our natural world to be revitalized and in some cases, retake lost habitats. But now, as humans tentatively re-emerge, we are faced with a war in Europe that further weakens the global supply chains, more so from an area from which a substantial percentage of our food supply comes. The consequence of this most recent challenge is that food security is now moving to the head of the list of challenges buffeting Trinidad and Tobago and the Caribbean and other small island developing states globally. So this evening we talk about microbes. We will hear from a speaker who is both knowledgeable and experienced in his field of study, who will tell us just why we need to take note of them, why microbes can be, as stated in his chosen topic, either savior or devil. Science tells us that microbes are essentially protectors of the planet. They ensure that minerals such as carbon and nitrogen are continuously recycled. They play a crucial role in keeping the atmosphere oxygenated and actively degrade dead organic matter so as to transform carbon back into carbon dioxide. A quick gander online will show proof that certain types of bacteria may be useful in our efforts to alleviate environmental health concerns, and health concerns, reducing global warming, cleaning up pollution, breaking down plastic, and perhaps even providing a cure for cancer. Professor Saravanakumar will in a few minutes explain why microorganisms play a critical role in the management of plant health by clearing the path to sustainable crop protection practices and eventually to food security. His presentation is yet another example of how the research we conduct here at the St. Augustine campus of the UWI as a whole will translate into practical, real-life solutions for this country and the region, and indeed the entire world. This evening is very much a landmark moment for Professor Saravanakumar, personally and professionally, as the inaugural Distinguished Professorial Series presents an opportunity for him to lecture and to share his achievements in research, engagement, in teaching, as well as his practical experiences in the study of this all-important area. The promotion of an academic to university professor, the highest academic rank, comes after an often agonizing journey of scrutiny of their teaching and research, first by our university community, and then once the candidate's application is found suitable, assessment of the candidate's body of work by a small group of international peers, expert professors in the would-be professor's discipline. Promotion to professorship cannot be granted until this group of professors who, even if they disagree with aspects of the theses espoused by the candidate, acknowledge that what is reflected in the candidate's CV and his or her publications demonstrates, and I'm quoting here, a record of distinguished original work, outstanding success in and wide recognition of professional activities, and enhancement of the reputation of the university in the field of work of that person through his or her own contribution to the development of the field in areas such as academic leadership, postgraduate studies, and other projects or activities. I'm taking an extract here from Ordinance 8 on academic staff promotions at UWI. It is through the achievements of academics, and this is very important, through the achievements of academics, teachers and researchers alike, such as Professor Saravanakumar, that the UE can boast of having achieved its current international reputation and its correspondingly enviable global top 1.5% university ranking according to the Times Higher Education Survey. What is not well known, or not as well known, is that according to the latest Webometrics statistics, Webometrics is another ranking agency, but according to Webometrics, the St. Augustine campus has been ranked number 1 among all other UE campuses. So I say thank you to academics like Professor Saravanakumar and others who have worked tirelessly over the years to bring us to this high level of recognition. Now we, his colleagues in the campus community and members of the public, will today learn from Professor Saravanakumar as we increase our understanding of the possibilities available to us to create more sustainable lives and livelihoods. Further, his family and friends with this evening share in his achievements and get a behind the scenes glimpse of what exactly an academic does, why they do it, and why those many hours spent in study and research were worth the sacrifice. Ladies and gentlemen, our speaker's journey to the UE has been a long one, not just academically, but also from a geographical perspective. Journeying as he did from India through Italy to Trinidad and Tobago, we are so very grateful that he made the trip. I congratulate him on his achievements thus far and extend best wishes to him and his family for the future. I thank you.
Thank you, Professor Copeland. It's quite a journey for any person who achieves professorship. The Dean of the Faculty of Food and Agriculture, Dr. Mark Woody Vera, will now introduce Professor Sara by way of his biography. Dr. Woody Vera.
Thank you very much, Professor Neela Badri, Chair, Open Lectures Committee, Professor Brian Copeland, Vice Chancellor and Campus Principal, Dr. Gaius Yudoxi, Senior Lecturer and Deputy Dean, the Faculty of Food and Agriculture, the moderator of this afternoon's proceedings, family and friends, ladies and gentlemen. I'm pleased to have the distinguished honor of introducing our featured speaker this evening, Professor Duraisamy Saravanakumar, Professor of Plant Pathology, whom we fondly call Professor Sara. Professor Sara is the current head of the Department of Food Production in the Faculty of Food and Agriculture. He holds a BSc in Agriculture, an MSc in Plant Pathology, and a PhD in Plant Pathology from the Agricultural College and Research Institute of Tamil Nadu Agricultural University in India. He joined the University of the West Indies St. Augustine campus as a senior lecturer in 2014. And prior to this, he held a range of academic positions at Tamil Nadu Agricultural University in India and the University of Turin in Italy. He has 15 years of experience in developing microbial-based novel formulations for the management of plant health, including post-harvest diseases. He is currently engaged in research on the development of molecular diagnostics of tropical plant pathogens and bioformulations for the sustainable management of plant diseases. He served as an international consultant for the Food and Agricultural Organization to develop the certification protocol to produce disease-free planting materials in ginger and turmeric in Jamaica. He has supervised 15 graduate students, with another 12 currently studying for their MSc, MPhil, and PhDs. Professor Sara has published over 60 impactful research papers in refereed journals and authored 9 book chapters. His publications have amassed over 3,000 400 citations, and with a whopping H index of 25 on Google Scholar. And papers that he has published as first author have earned over 1,500 citations. His publications are in the fields of plant pathology, entomology, biological control, stress mitigation, microbiology, environmental science, and molecular biology, amongst others. He has received externally funded research grants totaling over $5 million as principal investigator and key expert from funding agencies such as JEF, FAO, European Union, UE, Trinidad and Tobago RDI Fund, Campus Research and Publication Fund, and UNU, BioLac, United Nations University. Prof. Sarah holds professional membership in a range of professional bodies, including the American Phytopathological Society and India's National Environmental Science Academy. Prof. Sarah is also a topic editor for the Frontiers Journal on the topic of plant growth promotional microorganisms for sustainable agriculture. From the foregoing, you will agree with me that Prof. Sarah has demonstrated an outstanding excellence in research that is unassailable. His work has reached the wider section of scientific community in updating knowledge and research in the fields of agriculture, plant pathology, entomology, microbiology, biochemistry, food science and technology, environmental science, and molecular biology. His work has also impacted the farming community and private industry in the development of biological-based disease management strategies. His research has made significant advances in the sustainable management of plant diseases, with significant impacts on food production, food safety, environmental and human health in the Caribbean region and globally. The recognition of his research findings could be seen in the publications in highly reputed peer-reviewed journals and the voluminous Google citations they have attracted. This has drawn immense attention from the international community to the University of the West Indies as a leading research institution in the field of plant pathology. The quality and impact of his research has undoubtedly boosted the standing of the University of the West Indies among the elite universities of the world. I now welcome our erudite, accomplished, and outstanding academic, Professor Duraisamy Saravanakumar, as he robustly tells us about microbes, a devil or savior of plant health. Help me welcome Prof. Sarah. Prof. Sarah, over to you. Thank you.
Thank you, Dean Dr. Udivera, for that nice introduction and for your support and leadership and encouragement, not only as a dean, but also as head of the Department of Food Production. I'm so much thankful and grateful to you. Also, I thank Professor Neela Badri. She was the head of the department when I joined the department in 2014 for her continuous support and encouragement and leadership. Your support is tremendous and impeccable, Professor Badri. Thank you so very much. Thank you, Professor Copeland, campus principal, for joining this evening with us. I'm not saying this just for the sake of formality. Being head of the department for 1 year, every day I receive hundreds of emails, and I could imagine the number of emails and responses that you have to do every hour. So thanks for joining with us this evening. I also thank Dr. Yudaksi, our Deputy Dean, Outreach and Internationalization, for his acceptance to do the moderator of this session. Dear colleagues and friends, I know you are all here for me to support the lecture. I'm so much grateful and thankful to all of you. Let me share my screen. So this evening, I'm going to present a topic, microbes, a devil or savior of plant health. For the purpose of presentation, I know there will be different types and range of audience. To make everyone the best possible way to make you all understand what I'm going to present today, I have this organization of my lecture. First, I'm going to say about my background. Then we are going to say why microbes are devils to plant health, how to identify a savior microbe of plant health, how does savior microbe suppress pathogenic microbes in plants, how to test and develop savior microbe for use in commercial cultivation of crops. Finally, conclusion and acknowledgement. Look at my background. I come from a state called Tamil Nadu, and that's the state in southern India. I come from a simple and small family. My parents, they did not have a university education, but they wanted their sons to have that university education. For that, they have sacrificed a lot. My mom, she used to be in the farm every day. My father, he was in a government system as an office assistant. And this is our hometown, Bhavani Sagar, and which has one of the world's largest earthen dam, which has the river runs up to 201 kilometers, irrigating more than 200,000 hectares of agriculture. So this says that I'm born and brought up with the agricultural background. Coming to my schooling, I have completed my schooling in a Kasturiba Niketan High School. That is the school has the name of Gandhiji's wife. And there you could see me doing some talk on the Gandhian principles, one of his birthday anniversaries. Then I have completed my secondary education at Diamond Jubilee Higher Secondary School, which has completed 100 years of its existence in Gopichettipalayam. And there you can see I am standing there with my friends. Then I entered into a university education. That is Tamil Nadu Agricultural University, Coimbatore. Until I entered into the Tamil Nadu Agricultural University, I never realized that agriculture has so much of science in it, so much of technology in it, so much of policy in it, and so much of research in it. Thanks to Tamil Nadu Agricultural University, like how the Faculty of Food and Agriculture has been started by the Britishers long before as the Imperial College of Tropical Agriculture. Similarly, the Agricultural College and Research Institute was started in Coimbatore in 1908 by the Britishers. Then now it has completed its 100 years. I'm so proud to have my schooling as well as university education as well as my workplace, which has completed 100 years of its existence and serving its excellence to the stakeholders. There I completed my BSc Agri, MSc Plant Pathology, and PhD Plant Pathology. And you could see some of my friends join, and I have one of my friends from Canada who joined, and he works in a industry on seaweed extracts as well as on plant disease management. Then after completion of my PhD, I have been to the University of Torino, Italy, for doing my postdoc, where I did work on post-harvest disease management using beneficial yeast strains. I worked on apples, peaches, peach, kiwi, then pears. And it was a nice experience, having completed my studies from the subtropical environment. Entering into a tropical environment and temperate environment, it has given a very rich experience. Then in 2014, I joined the Faculty of Food and Agriculture. Since then, I'm enjoying the warmth of Caribbean. I've been collaborating with Guyana Rice Development Board and the Research Division Ministry of Agriculture in Jamaica, as well as we're doing work in St. Lucia, and also having collaboration with Food and Agriculture Organization of the United Nations. I'm so grateful and thankful for all those collaborators and the regional institutions that are collaborating with me. Coming to the topic, microbes, devils or savior to plant health. If you look at the plant-associated microbes, there are different types of microbes that exist. Either they do harm or else they benefit the plants. The microorganisms, fungal microorganisms, or fungal-like microorganisms, that is later regarded and identified as algae, that is the chromista, then bacteria, virus, and virides, and phytoplasma. All of these microorganisms, if you look into the history, then we can understand whether they have done benefit to the plant health or else they have done harmless to the plant health. Here, I have given the title, let us see why microbes are devils to plant health. If you look into the history, in 1840, there was a serious disease affecting the cultivation of potato that is called late blight. Because of that disease, 1 million people died due to starvation. 1.5 million people migrated out of Europe. Even before that, there was a disease affecting the cultivation of wheat, that's the cereal. People have seen and noticed reddish-brown spots. During that time, they did not realize that was a disease. They did not have a microscope. They did not have a knowledge of the microorganisms. They thought that is the God's curse to the human beings. They thought that is the God's punishment to the increased criminal and wicked activities of the human beings. So they collected red-colored animals like horses and dogs, and they sacrificed to the god to please the god. But that did not solve the problem. And that's how they have shifted their cultivation from wheat, and potato was introduced. After a few years of cultivation of potato, it has a serious disease, as I said, late blight, which is caused by Phytophthora infestans. Similarly, if you look into 1860s, there was a serious disease affecting the devastation of coffee plantations in Sri Lanka. And during that time, the Britishers, they introduced tea plantations and they become tea drinkers. This disease was a serious problem in Jamaica as well, started since 2012. Then in 1920s, we can see Fusarium wilt that destroyed Gros Michel variety in Costa Rica as well as in Panama. That's the Panama wilt of banana. And that disease has led to the development of resistance variety called Cavendish. 1943, there was another disease affected so many people in India that is called Bengal famine. 2 million people died because of that brown spot disease. In 1960s, again, bacterial leaf blight disease that caused serious yield loss, more than 80% of yield loss in Asian rice cultivation. Witch's broom disease, another fungal problem in cacao production in Brazil, which reduced 75% of the annual production. As I said, in 1920s, we have seen that the Fusarium wilt has started affecting the cultivation of banana. That was the race 1. The same pathogen has changed into some other genome structure, and that now it has taken as the TR4, that is the tropical race 4, which is more prevalent in Australia and Africa and Asian countries. It is not present in the region. So the region is trying to prevent the entry of this tropical race 4 to protect the cultivation of Cavendish, as Cavendish is also highly susceptible to this tropical race 4 pathogen. Papaya ring spot virus that wiped out the cultivation of papaya in Hawaii. There is another disease that affects the cultivation of wheat in Africa and Middle East countries. That is Uganda 99, that is UG99 race, which is known to affect the cultivation of wheat. Since we are talking about the introduction of some cereals in the region, and we have to be aware of this type of diseases as well. And also there is a bean rust affecting Asian bean rust, affecting the cultivation of beans in the region. So these are all some of the global diseases that caused serious loss to agriculture, and indicates that the microbes, they can do a lot of harmness to the plants. If you look into the region, the tropical weather conditions, that has more favorable conditions for fungal, bacterial, chromista, viral, and phytoplasma diseases. We have studied number of diseases in the region, not only in vegetables, but also in spices, as well as in cereals like rice. However, for the purpose of the presentation, I'm going to focus only on the vegetables, the work that we have done in Trinidad with vegetable diseases, as well as the management practices that we have developed with the Savior microbe. If you look into the vegetable production in Trinidad, hot pepper is one of the most important commercial crop with high export value. It can be exported to Canada as well as UK due to Caribbean diaspora, and it is one of the hottest peppers in the world. At the same time, if you look into the challenges that we have in the cultivation of hot pepper, is collar rot and wilting disease, which is again caused by Phytophthora. 40% of crop loss is noticed regardless of pesticide application. However, there is no formal study on the pathogen as well as the symptoms and the management practices. What we have done here, we have collected the samples that is expressing the wilt symptoms, and we have grown that in the lab. This pathogen is very difficult to grow them in the lab due to its biotrophic nature. When I say biotrophic, the microorganism can grow and multiply only in living cells. In lab conditions, it would be very difficult to cultivate unless otherwise we used a specialized media. We have used different types of media to isolate this particular pathogen, and we have studied the morphology of these pathogens from the isolates or from the samples that we have collected from Valencia and from Macoya. And here, if you look at this Phytophthora pathogen, it is known to produce two types of reproduction. One is the asexual reproduction, the other one is the sexual reproduction. Asexual reproduction normally happens with the microorganisms when they enjoy the nutrients, when they have the favorable conditions, and when they have the cropping seasons. That is the season where they go for asexual reproduction and produce more spores and spores and spores and spread to the plants and crops immediately. But when there is no food for the microorganisms, when there is no nutrient for the microorganisms, once the crop is going to the end of its life cycle, the microorganisms, they will stay quiet and they will take another route going into the dormancy, and they will go to the sexual reproduction to produce the stronger spores, which can remain in the soil for so many years. Microorganisms, to me, they are very, very intelligent creatures. If you look into this asexual reproduction, we have, though we have isolated several strains, here, two isolates, they had distinct characters. One is the mako one, the other one is the mal one. And here, the sporangia, this is the lemon-shaped one, which produces the spores. And inside you can see the juice spores, that is the kidney-shaped one, and they can swim in the water. That is why this type of disease can occur during wet conditions as well as during high rainfall conditions. There is another type of lemon-shaped sporangia produced by the another isolate called mal2. And this is the asexual reproduction, and we have characterized this pathogen. But what is interesting here, when we study the sexual reproduction of that particular pathogen, when we allow the individual pathogens to produce the sexual spores, they did not produce, they did not pair it. But at the same time, when we allow two of these distinct isolates to mate with each other, and here they produce the oogonium and antheridium, male and female gametes, and they fuse together and they produced the resisting or resting oospores, which can remain in the soil as a dormant structure for so many years. So this can produce the different types or the strain with high level of genotypic diversity. And this could also produce the strain which can develop resistance to the fungicides. This we have noted in one of our experimental trials, where the fungicides that has been exclusively used for the control of Phytophthora disease is not giving any control. It indicates that this isolate has developed resistance or has become insensitive to that particular fungicide. There is high possibility for developing resistance by this type of isolates. We have identified using PCR techniques, and thanks to COVID-19, now the global population has been very much sensitized about the PCR technique. And I need not explain anything about that. And PCR is a simple technique used to identify a specific, or amplify a specific DNA fragment from the genome of our interest. And here, the molecular identification of these two isolates using universal primers, as well as the cytochrome oxidase gene, that is the mitochondrial gene, has confirmed that they are the Phytophthora capsucei. Based on the DNA sequencing, we have confirmed, as well as the pathogenicity study also confirmed, that they are highly virulent strains. Then we have studied the diseases of next vegetable crop that was leading the production of vegetables in 2015 with the production of 2,800 tons. And after that, it has been reduced. In cultivation of lettuce, we have observed two important diseases. We have made a survey of 60 farms. Out of 60 farms, more than 80% of lettuce farms that we have visited had a disease incidence. That ranged from 20 to 80% in both field as well as hydroponic cultivation of lettuce systems. And as I said, two types of leaf spots. One is the light brown spots with gray center. The other one is oily and dark brown spots. We have assessed the yield loss from field to field, 20 to 40%. And also when we have conversed, or else when we have had a conversation with the farmers, we realized that 2 to 3 times they are applying fungicides in a week. Also, we have noted that overhead irrigation encourages disease spread. We were interested in knowing the pathogens that cause this disease. Though based on the symptoms, we will be able to identify what it is, but still the confirmation in the lab as well as doing the molecular studies, as nowadays there are so many strains spread and so many new pathogens are emerging. Therefore, it is important to study what is happening and how they are evolving, and is there any new pathogens emerging in the vegetable production systems. Here, the leaf spot with the gray center, though it appears Sporepora, we have isolated and we have confirmed based on the conidial structure, it is Sporepora. And this pathogen is also hemibiotrophic in nature. It is difficult to isolate and sporulate. So we have developed a specialized technique using the host extract, which again confirmed that this pathogen is specific to lettuce because it can grow when the lettuce extract is added to the medium. PCR amplification again confirmed the amplification of ITS primers as well as this DNA sequencing confirmed this as the Sporepora lacticase sativa. What is interesting here is when we grow this pathogen in the lab, the colony growth was enhanced at 25 degrees Celsius, but the conidial production was enhanced at 28 degrees Celsius, which shows that when these temperatures exist for a period of time, there is a high possibility that the pathogen could multiply more spores in a short time and it can spread to the other plants. The other Leafspot that we have noticed with grassy oil as well as dark brown spots from Lopinat, Cunupia, and Princetown, we brought it to the lab and we have isolated. In our isolation, we have noticed yellow color mucoid colonies as well as white color colonies. This yellow color colonies, when we have used the PCR, we were able to identify as the Xanthomonas campus trees. But these white colonies, they were appearing in all our repeated isolations. We may wonder how this has come. When we have inoculated both this yellow colony as well as white colony onto plants, both were able to develop the system. And here in this last picture, you could see the freckle leaf spots developed by this particular bacteria. When we have used the genus-specific primers, as well as based on the sequencing, we have identified one as the Xanthomonas campus trees, Pathova vitians, which was notified as a quarantine pathogen by the Ministry of Agriculture in 2012, but now it is widespread in the country. There is another pathogen which we have noted as an emerging one, Acidovorax valerinella. And this Acidovorax, when we looked into the literature, it has been reported in UK to infect lamb's lettuce. This raises the alarm that there are new pathogens are emerging because of the changing in weather conditions and tropical weather conditions. When new pathogens are emerging, there is a need for the sustainable management of diseases as well, or else continuous evaluation of disease management and protection practices. Also, we have noted another disease which is emerging in lettuce. We have noted wilting, basal rot, and soft tissue. We have brought this into the lab, and based on our study using PCR, we have identified two distinct genus. One is the Pectobacterium, the other one is Dickia. This Pectobacterium and Dickia, they have been identified based on multi-locus sequence typing using the PCR techniques as well as the analysis of the data. This Dickia and Pectobacterium, when they were inoculated onto lettuce, both were able to cause the disease. This indicates that this Pectobacterium can be more serious infectious compared to the Dickia based on our preliminary studies here. Ultimately, what happens when we have all of these diseases? The farmers will use the synthetic pesticides. When they use the synthetic pesticides, there is human, environmental, and food safety concerns. We always say that. If you ask me, do we have any empirical evidence to say it is affecting the human health or it is having a concern to the environment? The answer is yes. We have done survey in collaboration with Food and Agriculture Organization of the United Nations using the phone health app developed by Pan UK. We did a survey in 2021 with the 208 vegetable producers representing all counties of Trinidad. In that, we have noted and we have observed 14 persons from that population indicated that they have abdominal pain and bloody stools, and also the other type of gastrointestinal problems like vomiting. 22 persons, they reported they had serious respiratory issues like shortness of breath, and also they have the other respiratory problems, throat irritation, for example. Sensory and neurology problems also reported by 100 farmers. Some of them said they had serious headache within 24 hours of exposure to the pesticide. All of these 208 farmers, they have been involved in application of the pesticides. We have asked the questions specifically to note or capture what is happening to those farmers when they apply the pesticides within 24 hours, which usually known as the acute pesticide poisoning. Also, 100 farmers, those who use the pesticides indicated that they have serious effects on eye and skin irritation. All of this data shows that the pesticides, they have serious impacts on human health. The person incidence associated with the pesticides clearly says that there are highly hazardous pesticides that we use in Trinidad as well as in the region, because the same type of study also conducted by my colleague, Dr. Robinson in UWA Bona. It says that the person incidence associated with the pesticides in Trinidad, 3% of them had serious incidents, and moderate and mild, that put together 45%, and rest of the 52%, they did not experience the symptoms. It says that 50% of the population of the involved in survey indicated that they have serious concerns. Therefore, we need to look into this seriously. Therefore, the disease, when it affects the vegetable production systems, there is a yield loss, there is a food insecurity, there is an increased cost of production because of more pesticide use. One of the farmers who cultivate lettuce indicated that the fungicide cost, it has increased within a short period of time. Not only that, the residues that is present, especially in leafy vegetables like lettuce, it is a 6 weeks old crop. If there are about 4 or 5 diseases affecting the crop, if the farmers are going with 2 to 3 times application of the fungicides, 6 weeks crops, even if they apply for 6 times during the period, that is indiscriminate application, the pesticide residues are going to persist on the leaves, and they're going to pose threat to the human health. In addition to all of these issues, human health impacts on the environmental pollution. If we look all of this into a picture, certainly the pathogenic microbes are devil to plant health. Now, if we have a devil, and there should be some good force, that is the nature. If we have an action, there is an equivalent and opposite reaction. In our real life also, if we have a problem, each problem has a solution. But always the solution is hidden somewhere. We need to search for the solutions, and we need to identify where the solutions are hidden, and we need to research, and we need to bring it for use. Here, what we have done is, we have also searched for such good microorganisms, and we were trying to see, can it be a savior of plant health? Based on our research and based on our study here, we have identified a range of microorganisms. But for the presentation, I'm going to focus only on a specific type of microorganism. Look into the rhizosphere, that is the root region. In the root region, there are different types of microorganisms. However, specific group of bacteria which can attach to the root systems and utilize the metabolites produced by the plants, and in turn, it also helps the plant to grow better and better. We have been interested in those type of microorganisms. To locate those type of microorganisms, we have done an intensive survey to collect the rhizosphere soil samples of the different ecosystems of Trinidad and Tobago. When I say different ecosystems, it includes agricultural ecosystem and forestry ecosystem and coastal ecosystem. However, we have collected soil samples only from the ecosystems that had a virgin soil. We have isolated 200 strains of both fungal and bacterial cultures. Based on morphology and biochemical characters, we have identified the major group of organisms as Bacillus species. Based on the morphology as well as the biochemical characters, when we have identified as the Bacillus strains, then we have used a PCR technique to amplify as well as sequence to identify what are those Bacillus strains. When we looked into it, it has 7 different species based on our research. They are Amyloleucophasians, Subtilis, Cereus, Pumilus, Mycoides, and species. Then we have looked into the specific group of or type of genes, whether they are present in that group of microorganisms or not. Because these lipopeptide genes, they are known to be produced by the specific group of Bacillus organisms, which has antimicrobial, which is inclusive of antifungal, antibacterial, and antiviral and nematicidal activities. Our first set of screening was to identify those producers. If we have those producers, there is a possibility that they could perform very well in suppressing the pathogens. That is our interest here. So we have used PCR again, using the lipopeptide genes as the candidate markers to isolate or to screen those microorganisms out of 47 Bacillus strains. When we have used different lipopeptide genes as the candidate genes to screen those microorganisms, such as iturin lipopeptides and bacillomycin and fungicin primers and basilicin primers, surfactant and zwetamycin. Based on this PCR screening of all 47 Bacillus strains, we have identified that Bacillus amyloleucophasian strains, they were able to produce majority of these lipopeptides, or they showed positive PCR amplification to this Bacillus, to this lipopeptide genes when compared to other species of Bacillus. And here, Bacillus cereus, they produced some type of basilicin and fungicin. Bacillus megaterium, they produced bacillomycin and surfactant. And Bacillus pumilus, they produced surfactant and zwetamycin. It clearly says that the Bacillus amyloleucophasian, it has a potential to suppress the diseases through its antimicrobial activity. Then we have, when I say we, it is my students and myself. We have tested it against a range of pathogens affecting the vegetable production, like wilt and the lettuce leaf spot, and bacterial wilt affecting the production of tomato in the region. Then cholera, that is the Phytophthora, as well as Alternaria solani and Coleotrichum, that is known to cause the fruit rot in hot peppers and several other crops. When we have tested those, whether they are suppressing the pathogens in the lab conditions or not, and we have again noted that some of the Bacillus amyloleucophasian strains that showed positive reaction to the PCR amplification of lipopeptide genes, they suppressed the pathogens. But at the same time, you have to note that some of the lipopeptides producers, they suppress only some pathogens and they did not show high activity against all pathogens. And that indicates that some microorganisms which has the specific lipopeptide genes, that could be antifungal activity and some could be antibacterial activity, and some could show both the activities. Then we sequenced all of those 47 the microorganisms, and when we have grouped them into together, it was clearly shown in this phylogenetic tree. All Bacillus amyloleucophasian strains, they grouped together and they are identified as the major antibiotic producers. But what is interesting here, the primers that we have used to amplify the lipopeptide genes in Bacillus strains, that did not show any positivity to Bacillus subtilis. But still, the Bacillus subtilis grouped in this group, indicating that they have lipopeptides or other antimicrobial genes, and those antimicrobial genes may not be captured in our study. When we have done the further studies, we have identified that it has antimicrobial compounds as well. But what is interesting is still 16S rRNA gene sequencing was able to group these antibiotic producers together, and that can distinguish from the non-antibiotic producers. After this identification of antagonistic strains, our next ultimate aim is to identify whether they suppress the plant growth or promote the growth. If we have a good antagonistic, the biocontrol agent or else the bacterium, which can suppress the pathogen, but at the same time, it should not suppress the growth of the plants. It should promote the growth of the plant. Only then we will be able to use in the plant system. Here, some of the antibiotic producers, they suppress the plant growth, and those we have removed from our screening, and only we have taken forward the strains which has shown both the lipopeptide producers as well as which has shown antagonistic activity against the pathogens, as well as showed the growth promoting activity in the seedlings. This is how we have screened the strains. 3 different strains we have identified an activity in all of those our preliminary studies. One is the PRIN1, the other one is OG2, the third one is SHUG1-1D. And here, then again, we have tested all of these Bacillus amyloleucophaciens strains in 2 different vegetable production systems. One is the lettuce, the other one is the hot pepper. And also we have conducted the field trials. Here in case of lettuce, what we have done, we have inoculated or we have treated the lettuce plants with this Bacillus amyloleucophaciens strains. And also we have one treatment with the fungicide, the other one neither treated nor, not treated with any of the fungicides or the Bacillus amyloleucophaciens strains. Then we have artificially inoculated the Sporepora pathogen, that is the Leafspot pathogen, to see what's happening there. And we have assessed the disease index as well as the growth parameters using this standard plant pathology assessments. And in that, we have noted that this Bacillus amyloleucophaciens strain PRIN1 was able to suppress the disease as well as able to suppress the leaf spot lesions in lettuce plants that is treated with PRIN1 and challenge inoculated with Cercospora pathogen. Whereas in case of control, there was high disease incidence as well as the number of lesions was noted high, and yield was also high in case of Bacillus amyloleucophaciens strain PRIN1 and PRIN4 when compared to the control and fungicide. But at the same time, if you look into the yield as well as the disease suppression, this Bacillus amyloleucophaciens was doing the balanced effect. Then we have tested it in a hot pepper against collar rot disease. As I have indicated, those diseases we have characterized and those we have studied as a serious threats to the vegetable production. In this case as well, we have noted a specific strain called Bacillus amyloleucophaciens SHUG1-1D that suppressed the disease when we have inoculated the pathogen as well as treated the plants with this particular bacteria. But interestingly, here you could see the fungicide did not give any control on it. Coming to the tomato, we have noted another strain which could suppress the disease incidence when compared to the fungicide and control. In both of these diseases, if you look into it, the fungicide, it had no impact or effect on these diseases. It indicates that the pathogens would have developed resistance or insensitivity to the pathogen, to the fungicides. Now, how does it suppress the pathogen? For that, we have extracted the lipopeptides from the microbial cultures. We have tested it against the pathogens, null pathogens. When we have tested it, we have noted that it can lysis the conidia of the pathogen, as well as it can lysis the mycelium of the pathogen. Then we have used it for characterization using the HPLC techniques. Because of time, I may not be able to discuss all of those characterization studies here. We have also taken another route through genome mining for discovery of pesticidal and phytocarbonyl compounds. And in this approach, what we are trying to do, we are trying to identify the genes which can be responsible for antimicrobial compounds. We have identified based on the whole genome sequencing of our strains. These are all the compounds that could exist in those microorganisms. However, they could be silent in certain settings and conditions, and they may not be produced. So we wanted to take this approach of identifying those genes and expressing them, then identifying the characters and discovering the pesticidal properties, including antifungal, antibacterial, and antiviral properties. On the right-hand side, you have identified many plant colonization and growth-related genes. The same approach will also be applied, connecting genomic approach with the spectrometry approaches to identify these fungicidal compounds. That is one of the possibilities. We are looking into it, and one of my students is now doing that work. Now, apart from this direct suppression of pathogens by the lipopeptides, what happens to plants when both microbes are present? Both the pathogen as well as the savior microbe, here I would say the Bacillus amylolicofaciens, since it has some suppression against the pathogens. When we are allowing it to grow together, they are doing the suppression. But what happens to the plant? Is there any activity in plant? For that, we have taken a system, there is a hot paper. We have inoculated plants with Bacillus, and we have inoculated plants with Phytophthora. We have inoculated plants with both Phytophthora and pathogen. And also we have inoculated plants with just water to check what's happening there. We have collected samples at different intervals in like 0, 6, and 12 hours and 24 hours. And we have done the enzyme analysis on key marker defense genes. Plants, they have defense genes and defense activity when something or when some stranger tries to enter into the plant system. Like humans, when some microorganisms are entering, then the human immune system is activated. Similarly, in case of plants, they also have the defense mechanisms. They are also known to produce pathogenesis related proteins. Here, we have assessed 2 key marker genes, the enzyme activity, that is the phenylalanine ammonia lyase, as well as the peroxidase activity. These 2 key enzymes are known to involve in strengthening the cell wall of the plant cell, as well as they are known to involve in the production of several secondary metabolites that can possess antimicrobial activity, preventing the further entry of the pathogens into the system. When we have tried here, at 72 hours after inoculation, if you look into the treatment with biocontrol agent, that is Bacillus amylolicofaciens, that is inoculated with the Phytophthora expressed more when compared to other treatments. It indicates that when plants are inoculated with Bacillus amylolicofaciens and challenge inoculated with Phytophthora, the Bacillus amylolicofaciens is able to induce more accumulation of defense enzymes and proteins, thereby preventing the entry of the pathogens into the, or development of the pathogens into the plants. Based on that, we have collected protein lysates, and we have used this 2-dimensional gel electrophoresis to know what is happening to the proteins in whole of the plant system when they are inoculated alone with SHAGUAN1D, that is Bacillus amylolicofaciens, and Phytophthora, and inoculating both Phytophthora and SHAGUAN1D. When we have assessed, there were about 51 proteins differentially expressed. Out of 51 proteins, our interest gone to specifically 10 proteins because of its high expression as well as the different systems. We have noted that these 4 proteins, they have been upregulated. One is the mitogen activated protein kinase, which is highly related to plant defense mechanisms, as well as the patatin, that is another one, which is known to increase the defense mechanisms through jasmonic acid pathway and oxylipin pathway. And also there is a heat shock protein, which is highly expressed in the plants, that is inoculated with Bacillus amylolicofaciens and challenge inoculated with Phytophthora, indicating that the plants can induce these defense responses when they have the support of the beneficial microbes. Also, there are about 6 protein spots that we have characterized, and they have down-regulated in plants that is treated with Bacillus amyloleucophosins and Phytophthora, but they have upregulated in a plants that is treated only with Phytophthora. Those show that they are related to hypersensitive responses as well as stress-related ones. This indicates that the plants, they are allowing the infection of Phytophthora in a plants that are only applied with Phytophthora. That you can see here in the previous one, the disease incidence, if you look at it, in case of control plants, which is only inoculated with Phytophthora, that has high incidence of diseases when compared to the other ones. This indicates that the Bacillus amyloleucophosins strains could induce defense enzymes in plants that are treated with Bacillus amyloleucophosins strains. Now, all this academic research, the farmers or the layman question would be, so what? How these are going to be useful for the farmers? You are saying this protein has expressed and this gene you have identified, but what's the use for us? What we have done is we have conducted field trials, range of field trials in the field to assess what happens when they are applied in the field conditions. We have assessed the Sporepora as well as bacterial Leafspot, and we have noted that 70% of disease suppression in the field, as well as close to 30% increase in the plant growth, the yield. And here you could listen to the farmer.
So what you think? All here that start to get water mark. That's kind of sweat and you see the water mark. That thing I using like it kind of sticky, you know, or whatever. Okay. So when it land on there now, like it preventing the water from staining and damaging it. Okay. You know, like when the water hit it too much, it is blister. Well, we ain't getting that. Okay. I tell you, I cut about 5 this morning and I surprised nothing. And then he tell me don't cut from them much because he was trying to accommodate a customer. Okay. I still remember the man had to wait until he said he want to wait. So what you gonna do? You gonna left all them pay to wait. So when they get big now, you go see the correct size. But so far, as they show we to apply it, we miss a day. If we did miss a day with applying it the way you show us, we might have run into some problems. Just so I said to use it, how we use it. And no matter how we see it, we ain't using no fungicide. None. We have a special can for that. Like the blue can, only that alone we using. And we mix it no water. We use it with 2 bottles, just so, but as it get bigger, now we have to get more.
Yeah. So that is the feedback from the farmer saying that the formulations or the bacteria that we have given has been doing excellent in controlling the diseases as well as in promoting the growth. Then we have conducted an experiment with hot pepper that has shown a different strain, that is the Shagwan 1D, that has given a suppression to the diseases as well as increase the yield when compared to the control. And that was actually comparable with that of the farmer practices with pesticides. We have conducted the training to demonstrate with the farmer as well as with Food and Agriculture Organization of the United Nations. In addition, we have also conducted the trials in tomato to know how these formulations are doing in the field. Here, you could see the data shows that it cannot give 100% control to the disease as that of the chemical. But still, when we look into the economy of the chemical, cost of the chemical, as well as the impact of the chemical that could cause on the field, we could realize that even if it is not giving 100% control, it could be a very good or viable product for developing integrated management practices. And the yield, interestingly, due to its growth promoting activity, we were able to see it promotes good growth. Now, with all these field trials, we have to make that available in the market. To make it available in the market, the most important thing is you should have a good shelf life. if you're putting out in the market, at least it has to stay for 6 months. Otherwise, if the farmer buy it a day and tomorrow he has to use it, if you say that, no one is going to buy the product. So you should have a high shelf life. And the next thing is, it should be suitable for different delivery systems. And also you should have a good efficacy during that storage period. For that, we have tested different types of combinations to know what is the shelf life and how to improve the efficacy and how to develop the formulations for delivery into different systems like soil application or foliar application, or we wanted to do the seed treatment, or we wanted to apply there, which has to release the bacterium for so long. That is the slow release type. Based on that, we have developed 4 diverse type of formulations for these strains. And those has also been tested in crops like lettuce and tomato. It showed that you could see here the growth promoting effect of those formulations that has been developed when compared to the control, and different types of applications to the seed was applied here. That indicates that they can sustain their growth and they can stay. Based on that, we have developed 3 different products based on the strains, and we named it as Biophyte 1.0. Then the safety of those microorganisms. When we look into the safety of those microorganisms, it is very, very important. We are saying that we are developing something, but at the same time, it should give control and it should be safe. When we tested it in a cabbage, we have studied the insect diversity. And in that insect diversity, we were able to notice in one of the applications, you could see the picture on the right-hand side, one with the chemical application, and on the left, that is the Biophyte applied one. The insect diversity says that it is safe to non-target organisms. Not only that, we have conducted a clinical trials in collaboration with Vet School, thanks to Dr. Sundaram, for conducting the studies to assess whether the active ingredients as well as the products, whether they have any acute oral toxicity or pathogenicity, pulmonary toxicity or pathogenicity. Here we not only tested the toxicity, but also the pathogenicity being a microorganism. Also, we have tested for acute dermatotoxicity and eye irritation, and it clearly shows that they did not have any abnormality. All of them showed the normal cases and they did not create any irritation. That has given the confidence that the microorganisms and the formulations that we have developed are safe to the animal, and it actually can be replicated to the human health as well. Now, coming to the conclusion, pathogenic microbes that has the potential to cause serious outbreaks in the region, which could affect food security and food safety aggravated by climate change. As I said, there are tropical conditions which can cause outbreak of diseases. Not only here in the region, but anywhere in the world nowadays, the world has become global village. If something happens somewhere in the eastern part of the world, we are affected. Something is happening here in the western part of the world, the east is affected. It has been so close. We cannot say that we are in a safe zone. Therefore, it's very important to study and consider and how they are going to affect the region. And we need to take measures to put all those things in place. The next thing is plant microbes. If you look into the microbes, that has really got enormous potential for management of plant health. Coming to the question, whether it's the devil or savior, I would say microbes has got huge potential for saving plant health. But the only thing is we need to identify and we need to put strategically, and we need to identify strategically so that to suppress the pathogenic microorganisms. Like in a community or in a country, if we have a policing system in a strategic way, in a strategic manner, that will be able to reduce or suppress the criminals in the country. So similar to that, we should have the balance. And once we have the enough population of the savior microbes, we will be able to suppress the pathogens and able to save the plant health. With that, I really would like to thank so many of my well-wishers, supporters, collaborators, funding agencies, my teachers, professors, everyone. First of all, my students. Without them, I cannot reach this stage, be it in India or even in University of Torino, and here massively, if we look at it, I would say Augustus, Marisha Khan, Rajendra Prasad, Brian Smith, Neela, Mrs. Beckley, and Pascal, Johnny Smith. I'm not going to read all the names, but they are very important to me. Without them, I cannot do anything. And coming to the funding agencies or collaborators, of course, I'm so grateful and thankful to UWA RDA Fund, which supported me with 2 grants, wherein we have been doing a lot of other works as well, which I haven't included in the presentation. And Campus Research and Publication Fund, and Food and Agriculture Organization, Dr. Lopez and Mr. Maturin and Ms. Ramold. And also we have the other collaborators from the Ministry of Agriculture, Mr. Ramjetan and Ms. Sherwood from the Ministry of Agriculture, Jamaica, and Mr. Hasmut Ali from the Pesticide, Toxic Chemicals and Inspectorate. And from TNAU, I thank Dr. Ram Jagdish, who was responsible, or I would say he was the person who indicated that there is a position available here, by which I applied. And I thank Dr. Davide Speraro from the University of Torino. And also one of my students is now with Professor Anderson at the University of British Columbia. And also my colleague, Dr. Kandaswamy at Yale Canada Laboratories and Guyana Rice Development Board, Dr. Rajendra Prasad. I also thank campus principal, Prof. Brian Copeland, for all the support and the leadership that he has provided, and the encouragement. Our Dean, Dr. Woody Vera, thank you, Dr. Woody Vera. Your support is tremendous and is impeccable. Also, I would like to thank former heads, Professor Neela Badri and Professor Stone, former deans, Professor Ganpath. He is involved in all of these projects and he has contributed a lot. I'm so thankful and grateful to you, sir. Dr. Beckley, Isaac Beckley, Prof. Ganpath, they were responsible for the pathology lab that we have established, and the Vice Chancellor, Prof. Beckers, Professors Hari, Haran, Jaya, and Ram Subak, and Chaudhary, Doctors Ayub Khan, Dwight Robinson, Venkatesan Sundaram, and Krishnamurthy, and Dr. Kadiravan Gopalan. For everyone, I would like to specifically thank Mr. Bayer and Mr. Sampson for all the help that they have provided in the faculty, and to all of UWA family. Also, I thank my supervisors and professors, Professor Samyapan and Professor Bayan, as well as my school teacher and friends from my school in India, as well as university in India, and also friends in Trinidad. I thank all DFP and FFP staff, all my academic colleagues, technical and supporting staff from the lab and administrative staff, Ms. Bannister, Ms. Bijmohan, Ms. Maharaj, everyone, Ms. Antoine, all of you, and also University Pew Station staff for this support. Finally, I thank my family, my wife, Ramya, and my kids for the support, and my appa, that's my father, and grandpa and grandma, who actually taught me the preliminary agriculture when I was a young boy, and my brother, who is there for me at all times, supporting me, and friends and relatives, and my amma. She actually passed just before I became a professor. So thank you. Thank you all.
Thank you, Professor Sarah. Quite an interesting presentation. And we know that microbes are quite diverse and versatile, and we can screen them for many uses. And in this case, for plant health and for biopesticides, but they can also be applied, like in the case of food, for wines and yogurts and so on. And in the case of medicine, like for hormones and antibiotics, and even in the environment for bioremediation. So quite an interesting presentation. As we move on to the question and answer session, please ensure that you place your questions on the chat so that they could be answered by Professor Sarah. The question and answer session will be moderated by Dr. Gaius Eudoxy, who is a senior lecturer and deputy dean of outreach and internationalization here at the Faculty of Food and Agriculture, the University of West Indies, St. Augustine campus. Dr. Eudoxy, welcome.
Thank you so very much, Chair. A pleasant good night to everyone, particularly our participants, and as well our distinguished Professor of Plant Pathology, Professor Duraisamy Saravanakumar. And I have to admit, the salutations are coming in fast and furious in the chat, and it is so nice to see. And we do know that there are also some questions that are coming in there. So we would not delay any further. We will move straight into our question and answer session. And I want to extend my thanks certainly to the participants for being so engaging and interactive. So I think, just as a preamble, while some of us may not be able to pronounce all of these scientific names for those microbes, I'm certainly, I think you would agree with me that we are in a position where we are now could better understand their importance within our food systems. This was a truly impactful presentation by our distinguished professor. Professor, the first question is a question that relates to the tool that was used during farmers' survey. I think that was the one with the FAO. So one of the participants asked, can you share a little bit more about the assessment or tool or framework that was used to conduct the pesticide study?
Okay. Thank you, Dr. Eudoxy, for that question. Actually, the tool used was Phone Health app that was developed by Pan UK. That was actually, they had a template, and that template was discussed with us, and we have provided the local settings and conditions to them. And based on that, that was fed into the app. And that, based on that app, we have students going to the farmers. And before that, we have identified the farmers, those who are using the vegetable production systems. And then the data was collected and it was analyzed based on the data collected statistically. And in addition to that, the Phone Health app itself, it captures all of these informations in addition to the identification of highly hazardous pesticides based on the information provided by the farmers. And then both were compared, and based on that, the information was prepared and the report was done. That is the information, but if they are specifically interested in that app or in that information, I will be able to share the information. We have done a presentation to the pesticide registers board, and the information can be made available to them as well. They also showed more interest to work with the other Caribbean islands as well to capture this type of information.
Thank you for that response, Prof. And I would encourage our colleagues and participants to please reach out to Prof at the department's office if you want to follow up. The second question asks, would you expect climate change to result in mutations of either savior and or devil microbes?
Dr. Eudoxy, can you please repeat that?
Certainly. So the question really asks the impact of climate change on the mutations that might occur in those microbes, and whether or not that might have an influence either on those that are pathogenic or those that are beneficial.
Okay, so with respect to the tropical systems, the pathogenic microbes with respect to the climate change, there is a possibility that the new microorganisms can evolve, and the minor microorganisms, they can become a major pathogenic diseases. But at the same time, based on our experience, these beneficial microbes, they can be a resilient microbes, and some of the microorganisms, they have the mechanisms to resist against the drought conditions as well as stress related conditions. So in that case, with respect to the climate change, these beneficial microbes, that they can be a resilient one compared to the pathogenic microorganisms.
Wonderful. Thank you. Thank you so very much, Prof, for that response. And we are hoping that our savior microbes outperform our pathogenic ones, you know, with the vagaries of our climate. Prof, with respect to farmers who are adversely affected by pesticides, did these farmers use appropriate protective, personal protective equipment? Did they follow manufacturer recommendations in the correct application of the pesticides? And I'm thinking again that has a lot to do with the survey that you conducted in Trinidad. Please.
Yeah, so we have captured that information as well. That's a very interesting question. When we have looked into that information, and only 24% of the persons, they have used personal protective equipments. And the rest of them, they did not use the personal protective equipments. And most of them, they have used just the regular class to spray the pesticides. And also when we asked about the training in the use of personal protective equipment, and that is also very low, and that was captured in our survey. That indicates that we need to create or we need to have more training to sensitize, as well as to ensure that the farmers, they are aware of these personal protective equipments and they use properly, so that the impact of those pesticides can be reduced on the farmers and the pesticide operators.
Thank you for sharing your thoughts on that, Prof. And as a follow-up question, the participant asked or indicated that they recently did a survey of new farmers' experiences in the use of botanicals. All the farmers noted they mix the traditional pesticides. Can you address the challenges of translating this technology to farmers' fields? And I'm thinking the technology here is the use of the botanicals.
Okay, with respect to the use of botanicals, I would say it is a good one, because it has been identified with a lot of antiviral activities as well as antifungal activities. But the challenge is where we are going to get all those botanicals. That is the question. Suppose if you are going to produce those botanicals, then again we have to grow those plants and we need to acquire those plants to produce the compounds. Once we are going to use those botanicals to identify the active compounds, and we are going to use that formula to synthesize the chemical compounds, that is fine for research. But if you are going to use those botanicals directly in the control of diseases, what will happen means, then again, we need to get that mass of botanicals. If they are freely available as a weed, for example, in case of seaweed extract, and we have that type of botanicals, and that can be used very well, because that is going as a waste, and that has the potential. But if you are going to use a specific crop, then we need to grow that crop as well, and that itself will take a toll on it. When it comes to the, for example, in case of microorganisms, and that can be grown in the lab and multiplied within no time. So in that case, the botanicals, if they has abundant and that can be used, it is very well, and it has the potential, and it can be used in the sustainable management of the diseases. But at the same time, we need to look into it where we are going to get those things. And based on its availability, it can be very well used in the disease management or pest management systems.
Thank you, Prof. I don't know if I'm going to put you out on a limb, but I also gathered from that question that there was some inclination towards, probably from an extension perspective, technology transfer perspective. What are your thoughts pertaining to how can we shift farmers' perspectives surrounding the use of those probably safer technologies rather than the traditional approaches with regards to chemical uses of pesticides?
With respect to that, the extension offices, we need to train, and the information has to be carried out to the farmers, and we need to, because nowadays it is a lot of information is not transferred to the farmers. And in that case, we need to use all platforms, and we need to educate the farmers and educate the stakeholders. For example, before COVID, no one knows about the PCR or real-time PCR or the coronavirus and its survival and how it affects the population and all of these things. But nowadays, if we go and talk to the common man, and common man is able to tell about the principles of the PCR and what is qPCR. And everyone, before COVID, mostly the plant scientists, even the medical researchers, they actually have been more into this PCR systems after this coronavirus outbreak. So what I'm trying to say is we need to educate and we need to make aware of the people about all these systems and how does it work. For example, in case of these microbial formulations, some of the farmers, and they are actually mixing the chemicals with the microbes and they are actually spraying it. That will have that is a serious challenge. So, but it can be rotated. It can be applied, the biological microorganisms can be applied. And after some time, the pesticides, it can be applied because the microorganisms that they will be able to survive in the system. So that type of integration is possible. But at the same time, mixing both microbials as well as the pesticides will have no effect. But I have noted that many of the farmers, without understanding or without knowing what is there as an active ingredient, they have been mixing and they have been applying. And that will have a impact. That will have no impact on the disease control. But at the same time, it will increase the cost of pesticides. If you are using it and if you have no control on it, then there is a investment in the pesticides. So the more technology transfer as well as the information, that will actually help the farmers. They're ready to accept the technologies. Once they are working, they are fine with it.
I'm happy that you indicated the importance of, I guess, directed as well as public education pertaining to understanding the modality by which those technologies work. And it is very clear from your presentation and from some of the responses by the participants that they are in a position of understanding of those. So the next question is follows on as well, and it asks, are these formulations commercialized? Because at this point, you probably have already convinced persons of the importance and impact that those beneficial microbes can have. So please, Prof.
So actually we should have been commercialized and made it available by this time. Because of COVID restrictions, there was some delay with some toxicology studies. Since we have completed all of these studies, now it is ready for registration, and we are in the process of going with the registration. Once it is a pesticide, we have to follow all of those protocols, and that's what we are doing it. And once also we develop here, then we have to generate a lot of data, which we have sufficient data to go for the registration with the Pesticide Control Board, and also would like to have it with the industries involved in this and make it available in the region as well. And not only that, the formulations that we have developed, we have given it to some of the farmers beyond the experimental trials that we have conducted. And we make those things available from our lab as a sample test. And they have also come out with some good feedback. And now one of the farmers is actually doing some mini trials in cacao production. The other farmers, we have given it for in tomato as well as in other crops like corn to know how this has been working and whether it could be extended for other crops and other diseases as well.
Prof, I know that you indicated that the option was taken to register the product as a pesticide. Can you elaborate why as a pesticide versus as a microbial biostimulant?
Okay, so here when we claim something as the disease control agent, then that has to be registered under the pesticides. Otherwise, we may not be able to advertise or promote that as the pesticide. So in that case, it has to go to the Pesticide Control Board, and it will be registered under the biopesticides.
Wonderful. Thank you so very much. Interesting question on cacao. Blackpot disease of cacao in Trinidad is a serious constraint to cocoa production. Would you consider delving into research on biological control of cocoa diseases in the future?
certainly that is one of our ideas, and one of my MSc student has done some work with isolation of trichoderma from the native soils to see or to assess its impact or activity against Phytophthora porta pathogen. We have done some preliminary work, but we haven't reached to the advanced level. We are planning to do it with the existing or the currently developed microbial formulations with some of the fungal antagonists as well. Not only the bacteria, we also have some of the fungal microbial agents which can suppress the pathogens. So we are in the planning process to extend the currently developed microorganisms as well as the strains that we have with the lab to test it against these pathogens affecting cocoa production.
Excellent. Happy to see that work is expanding to commercially viable crops, such as our cocoa. Another question, are there any interaction effects with keeping fungal and bacterial balance in the soil and fostering nematode growth? Not too sure if you appreciate what the question is asking.
Okay, with respect to the nematode growth, nematode is one of the main culprit to facilitate the entry of the fungus and facilitate the entry of the bacteria into the plant. So the nematode, some of these bacillus strains in the literature, if you look into it, some of them, they have the nematicidal properties as well. But to keep the balance of the fungus and the bacteria, the pathogenic bacteria, if we have to reduce, then the nematode population has to be suppressed. There are some cultural practices which can be used to reduce the nematodes, but still the nematodes are a serious problem. And there are some microbial agents like Bacillomyces that could be used for the nematode control. And such type of microbes can be explored for the control of nematodes.
Wonderful. It seems the beneficial microbes and their possibilities are boundless. Is it likely that plant pathogens can develop tolerances to these new compounds or to these new microbes?
Dr. Irasi, I did not get that, sorry.
So the question is, Prof, is it likely that the plant pathogens that are suppressed by the microbial agents, can they develop some level of tolerance against the mechanism by which they are being suppressed?
Okay, that's very, very important and interesting question, because the pathogens, they develop resistance against the fungicides. So in that case, what happens to the microbial agents as well? In this case, based on that, the resistance has not been reported. The reason is the biocontrol agents, or else these beneficial microbes, they have different types of mechanisms. One is like direct suppression. The other one is mediating resistance through the plants, and also mediating or the production of lytic enzymes. Since there are different mechanisms available against the pathogens, there is no direct resistance developed against the antagonistic microorganisms. But however, there is some sort of inconsistency noted with the application of the beneficial microorganisms. Then again, that is related to its persistence and existence of that microbial population, or its adaptability to that environment. Once we introduce into the microbial population into the rhizosphere, it should be able to survive there. If it is not going to stay there, then all these mechanisms is not going to work. So we need to ensure that the formulation or whatever we are developing will help or protect those microorganisms that we are going to introduce into the environment, can stay there, and only then it can do the work. Like very similar, if you have a policing system, and the police should be there where some criminal activity is taking place. If policing system is not there, even if we have a policing system, then that is not going to work. So very similar analogy here. The microorganisms that we introduce, it should be able to multiply, stay, and only then it can suppress the pathogenic microorganisms.
Thank you very much, Prof. A follow-up question. in line with the previous one, do the new microbial treatments propagate themselves in the soil and on plant and or on the plant? And after it is sprayed, or does it continuously have to be sprayed? So, for instance, what is the directions for use of these microbial agents?
Okay, with respect to these microbial organisms, it has to be sprayed for the vegetable systems. In our study, we have noted that every 2 weeks fortnightly interval, when we spray this is able to suppress the pathogenic microorganisms in an effective way. But here, the advantage of using these microorganisms, it is not only suppressing the plant pathogens, but also it is going to promote the plant growth. So in that case, it has to be sprayed in 2 weeks time. In case of perennial systems, at least once in 30 days, it has to be applied so that it can promote and it can keep the pathogenic microorganisms under the control or away from the rhizosphere system. It is not that once we introduce into the system, it is going to remain there for long. Once it is introduced, because there will be a lot of agricultural practices that we will be carrying out. So in that case, once we are going to do that, then that is going to affect that balance. So in that case, the repeated application is necessary. But again, the repeated application is not like pesticides. In case of pesticides, it's going to suppress the pathogens. And here, it is going to suppress the pathogen as well as going to promote the plant growth. In that way, it is going to take the advantage.
Thank you so very much, Prof. I have a combined question here by two participants pertaining to probably a timeline, expected availability of the product, and a retail cost, if that information is available at this particular point in time.
We have actually worked out the cost, and we've said for 1 gallon about $150 to $200. That's our cost. We have actually worked out the reasonable cost based on the product developed. In addition to that, the timeline, I'm really working with just now we have completed all these clinical trials and we have the data, and it will take certainly 2 to 3 months. We have apprised the Registrar Pesticide Control Board, and he is fully aware of the work that we have been doing. I hope we will be able to bring that out in the market, but still we are actually making it available from our lab through our faculty services. Those who are interested, they can contact and we can provide the product, but we will certainly trying to make it in 3 months' time.
Excellent. That must be very good news for a lot of our producers in this particular region. And noting the region, are there any avenues available for collaboration with other Caribbean countries? It was specifically asked about Guyana, but I'm extending it to the region.
No, certainly we are a regional institution, and we will be more pleased than anyone to have that type of collaboration. One of my students is from Guyana Rice Development Board, and we are always open. And Dr. Yudaksi is the Deputy Dean Outreach and International Station. So you could contact any one of us, and certainly there is a possibility, and we are ready to collaborate as well as enable you to do the same type of work in your island as well, and develop and discover more microorganisms that could be helpful to the plant health and agriculture and the environment.
Thank you, Prof. A question asks, research has shown that scorpion pepper has antimicrobial properties. Would you expect a synergistic effect on the pepper plant with your Bacillus bacteria?
We haven't tested that. We haven't tested that. I'm not sure whether it will have a synergistic activity or antagonistic activity, but there is a possibility. In some formulations, the botanicals, it can be combined with the microorganisms as well, and that can be used. So if we have a specific antimicrobial compound from the plants, and that can be utilized, and it certainly should have a synergistic activity. But what we have to know is, when we are developing a formulation, we need to make sure that whatever we are using it from the plant, that is not going to suppress the microbial organisms that we are interested in. So that compatibility have to be checked. And once that is there, there is no problem.
Wonderful. Prof, 2 more questions. I will leave one which is a little more philosophical towards the end. And I would ask, considering the nature of our farming systems, how important is it to also ensure that other cultivation practices are optimized for the effectiveness of the microbial agents? So things like fertilization and ensuring adequate water. How important are those practices in relation to the use of microbial agents for disease management?
Yeah, so with respect to that, the microorganisms that has to be introduced from the beginning, from the seedling preparation as well, from the seed treatment as well. But when it comes to the farming operations, there are amendments like the compost and the soil amendments, which always tend to help and promote this type of microorganisms to survive in the soil. So the soil health is also very important to promote this type of beneficial microorganisms and suppress the pathogenic microorganisms. So in that case, the application of organic amendments is very important. And the other one is the crop rotation. That is also an important process to promote the microorganisms, even if they are not going to promote the microorganisms, but at least to suppress the microorganisms that can be specific to tomato plant. For example, if you are going the cultivation of tomato every other year in the same field or every other season in the same field, then the nutrients going to be depleted and the pathogenic microorganisms is going to remain in that soil for so long. In that case, the crop rotation is another practice that has to be looked into it. And with respect to the fertilization, the nitrogenous and other fertilizers application that can help these microorganisms as well to take some of the nutrients. But however, applying some biofertilizers can be amended to promote or to have some synergistic activity with this type of formulations.
Thank you so very much, Prof. The last question, have you developed a specific philosophy towards the use of microbes for plant protection?
Specific philosophy, okay. So to develop microorganisms, it is actually to protect the plant, and they are again caused by the microorganisms. Then in that case, if you have the same type of microorganisms on the opposite side, then the balancing of these things, and that will actually help to outcompete the pathogenic microorganisms. And by that way, it can suppress the disease, and it can outperform the pathogens. And that's how I see it. And I don't know whether I answered this in a full-fledged manner. Thank you.
So that's probably we'll provide you with the individual who asked the question, and you can probably have a follow-up discussion on that. But certainly, I would like to thank the audience for a very interactive and engaging session with you. Plenty of questions, a few of them we weren't able to address, but I will ensure that I provide you with those questions, and probably there's an opportunity for that discussion to continue. So with that, I would like to offer thanks to our distinguished professor for gracing us with his presentation on microbes. And I think at the end of it, we can be convinced that microbes are saviors rather than devils. So I hope that we have all enjoyed that presentation. I would like to express thanks to the chair of this particular session, as well as our chair of the UK Open Lectures Committee, Professor Neela Badri. I would also like to express thanks to the organizing committee and those in the background supporting the efforts of that particular committee. Special thanks to our campus principal, Vice Chancellor, Professor Brian Copeland, for his opening remarks, as well. Special thanks for the introduction provided by our Dean of the Faculty of Food and Agriculture, Dr. Mark Woodivira. I also want to extend thanks to all of our staff and students who have joined us, our specially invited guests, media, and most important, our audience and our participants. Without you, this day of celebration of our distinguished professor would not have been as good as it was. So with that, we thank you so very much for your support. We ask you to please look out for the next distinguished lecture series. And with that, we will wish to bid you good night. Take care, everyone, and goodbye.